Hey guys, I'm your host Prisha, and welcome to the second episode of the Wellness Lab podcast. This podcast is a dynamic platform where students discuss the pros, the cons, the hot takes, and the ethical issues faced by up-and-coming science projects around the world. In today's episode, I'm here with Josh, Ryan, Shahzad to talk about the revolutionary gene editing tool that is stirring up quite a lot of curiosity in the scientific community. We're talking about CRISPR technology. CRISPR technology, i.e. clustered regularly interspaced short palindromic repeats, was in 2005 discovered as an immune system and now has the potential to bring scientific advancements in various fields, including but not limited to science, artificial intelligence, and de-extinction.
Today, I'll be talking about how CRISPR can be used to enhance stem cells, which can offer groundbreaking potential for treating genetic disorders, degenerative diseases, and advancing regenerative medicine.
In my segment, we'll discuss how artificial intelligence enhances CRISPR's precision while highlighting dangers like off-target effects and unpredictable long-term consequences. And in other segment, I'll be talking about the ethical issues, including germline editing, designer babies, and access inequalities will be examined.
With the rising potential, there are many concerns for social justice and physiological effects are important points of discussion.
Today I'll be talking about, first of all, how can CRISPR be used in the different fields of research such as medicine, agriculture, and bioengineering, and also what are some of the harmful side effects of it.
Now that we have acknowledged the and risks, let's begin the episode by diving into the impacts. We are going to be starting off by talking about type 1 diabetes. Type 1 diabetes, once known as juvenile diabetes or insulin-dependent diabetes, is a chronic condition. In this condition, the pancreas makes little or no insulin. Insulin is a hormone that the body uses to allow sugar or glucose to enter cells to produce energy. Different factors such as genetics and some viruses may cause type 1 diabetes, although type 1 diabetes usually occurs or appears during childhood or adolescence, it can develop in adults. Even after a lot of research, type 1 diabetes has no cure or reason for diagnosis. So the treatment for it is directed toward managing the amount of sugar in the blood using insulin, diet, and lifestyle to prevent complications. Let's move on to the symptoms. Type 1 diabetes symptoms can appear suddenly and may include feeling more thirsty than usual, frequent urination, bedwetting in children who have never wet the bed during the night, feeling very hungry, losing weight without trying, feeling irritable or having other mood changes, feeling tired and weak, and having blurry vision. The exact cause, as I mentioned, of type 1 diabetes is unknown. Usually, the body's own immune system, which normally fights harmful bacteria and viruses, destroys the insulin-producing islets or cells in the pancreas. Other possible causes include genetics, exposure to viruses, and other environmental factors. Before I tell you about the scientific advancements made towards finding a cure for type 1 diabetes, let me tell you about its main impact on one's life. Type 1 diabetes is a huge socioeconomic burden. Type 1 diabetes mellitus has proven to be a financial burden alongside providing physical and mental distress. The development of new technologies to cure and/or prevent such a disease would lift the burden from families and allow children to live long and healthy lives. The development of gastric inhibitory peptides, amylin analogs, and incretin mimics, alongside potential diabetes drugs including PPAR and DPP4, gene therapy has also proven to be quite effective in the treatment of diabetes mellitus. Speaking of gene therapy, CRISPR or Cas9 has led to the development of cutting-edge tools that can be used to inhibit or activate gene expression in a targeted way. CRISPR or Cas9 technology allows examiners to edit mammalian genomes in a certain way. It has been a building block in the development of high-tech tools that could activate gene expression as needed. This technology can be used to identify underlying diseases, correct mutations, and develop diabetes treatments. ViaCyte and CRISPR Therapeutics have come together to conduct a study which evaluates the potency of a stem cell therapy named VCTX210. This therapy was brought about to produce insulin for patients who suffer from T1D. VCTX210 comprises of allogenic pancreatic endoderm cells, which have already been edited by CRISPR-Cas9 technology to improve immune evasion and survival. Furthermore, the beta cells, which are responsible for the secretion of insulin, can be regenerated using transdifferentiation of stem cells, which will have a direct effect on the function and morphology of the beta cell's functionality as well. Adeno-associated viral vector-based gene therapy has also improved long-term glycemic control and prevented secondary complications in diabetes patients. Although we do not know the exact cause of type 1 diabetes, from as early as 2010, genetic mapping and gene phenotype studies have uncovered 10 key genes associated with type 1 diabetes, including those responsible for encoding major histocompatibility complex, or the MHC, human leukocyte antigen HLA class 2 and class 1 antigen presenting molecules, streptoinsulin INS in the thymus, cytotoxin T lymphocyte antigen 4 CTLA4 in dreg cells, and cytokines such as IL-2, IL-7, and IL-10, which are all believed to be causative factors in the development of T1D. The cellular and molecular pathology of T1D indicates that it is primarily an autoimmune disease driven by T cells. Studies have also found a connection between a rapid degradation rate in the immunoproteasome and inflammation, leading to autoimmunity and the destruction of pancreatic beta cells mediated by autoreactive T cells. The AID or RAD51 axis. Researchers developed an activation-induced cytidine deaminase AICDA gene knockout node mouse module using CRISPR or Cas9 technology. B lymphocytes from wild-type node mice showed high levels of constitutive AID expression, while ablation of AID led to significant delays in type 1 diabetes development but did not change the overall diabetogenic activity. However, combining inducible AICDA ablation with treatment with RAD51 inhibitors expanded the CD73 plus B lymphocytes that exert regulatory activity, which effectively inhibited diabetogenic T cell responses. The AID or RAD51 axis has subsequently been recognized as a potential target for approach with potential clinical translation. This approach aims to impede the onset of type 1 diabetes by transforming B lymphocytes into a regulatory state characterized by the disease inhibitory CD73 plus phenotype. CRISPR or Cas9 technology was employed to selectively inactivate two specific genes encoding protein tyrosine phosphates, phosphate Cs, in beta cells and islets. This genetic modification allowed researchers to investigate the roles of these proteins in the regulation of cytokine signaling and beta cell death in the context of type 1 diabetes. Finally, proponents of this therapy must also consider the treatment's accessibility. About 8.4 million people, half the population of New York actually, have type 1 diabetes. CRISPR or Cas9 treatments can be costly, so policymakers and insurance companies must ensure equitable access to treatments for such a common disease so as not to worsen existing healthcare disparities. I would like Josh to tell us about how CRISPR can enhance stem cell therapies.
CRISPR allows us to precisely edit genes, which is key to correcting harmful mutations in stem cells. This can be life-changing because when a mutation is fixed in a stem cell, the healthy stem cell can multiply and create a whole new population of healthy cells. These healthy cells can then replace damaged ones throughout the body. For example, if we were to correct a mutation in blood-forming stem cells using CRISPR, those edited cells would give rise to an entirely new healthy blood cell population. This is important because many genetic diseases lack a true cure. Most treatments simply manage the symptoms, but by using CRISPR to correct mutations in human-induced pluripotent stem cells, we could potentially produce healthy tissues that permanently replace damaged ones. Now I'll be talking about enhancing regenerative abilities of stem cells. CRISPR can also be used to boost the regenerative abilities of stem cells by modifying genes that control cell production, survival, or differentiation. CRISPR can enhance how stem cells work. This could be particularly useful in treating degenerative diseases like spinal cord injuries or Parkinson's disease, where cells are damaged and need to be replaced. Additionally, CRISPR can make stem cells more resistant to factors that reduce their survival, allowing for better outcomes in treatment. This matters because if we take Parkinson's disease as an example, CRISPR can help stem cells produce dopamine-producing neurons more effectively. This could be critical for restoring brain function in patients. Essentially, we're not just stopping the degeneration, but we're also actively repairing the damaged tissues. Now I'll be talking about combining CRISPR with stem cell transplants. CRISPR can be used alongside stem cell transplants to treat genetic disorders. For instance, stem cells from a patient's bone marrow can be edited with CRISPR to fix mutations that cause blood disorders. Once edited, these cells can be transplanted back into the patient, potentially offering a long-term cure. This is important because stem cells are often limited by donor availability. But by using the patient's own stem cells, edited with CRISPR, we can bypass this issue and offer a personalized treatment. This opens the door to efficient, individualized cures for various genetic disorders. We can now move on to Shahzad to tell us about artificial intelligence in CRISPR.
Let's dive into the role of artificial intelligence, or better known as AI, in CRISPR technology. These two powerful tools are transforming the way we approach genome editing, drug development, and even personalized medicine. But how exactly is AI helping to shape the future of genetic research? Let's break it down. One of the most critical aspects where AI plays a significant role is automating and accelerating data analysis. CRISPR experiments, especially those involving large-scale genetic screenings or whole genome sequencing, generate loads of data. Manually analyzing this would take weeks, if not months. But with the use of AI, the process becomes faster and more efficient. For example, AI can automate the analysis of sequencing data, helping researchers identify successful genome edits while also flagging potential off-target mutations. By automating this, it reduces the labor-intensive work, allowing researchers to quickly validate results and keep refining their experiments. AI isn't just about speeding things up, it's also expanding what's possible. Most of us are familiar with the popular CRISPR-Cas9 system. But did you know there are many other CRISPR systems? Systems like Cas12 and Cas13 can target RNA instead of DNA or are smaller and easier to deliver into cells. Through AI-driven bioinformatics, machine learning models can sift through enormous genomic datasets, helping us discover new CRISPR systems. With unique capabilities, imagine the potential of finding a CRISPR system that can work in a completely different way. AI helps us open the door to these discoveries. By discovering novel Cas proteins that might have different cutting mechanisms or target preferences, AI expands the toolbox available for genome editing and opens up new possibilities for research and therapy. Let's talk about CRISPR in therapeutic development. We're moving closer to personalized medicine thanks to AI's ability to integrate patient genetic data. With CRISPR, AI can analyze gene functions or even identify potential drug targets by systematically knocking out thousands of genes. So what's the result? We can speed up the discovery of targeted therapies and more importantly, create personalized treatment plans. By predicting how specific genetic edits might impact a patient, AI allows us to tailor therapies based on their unique genetic makeup. Beyond just research, AI is streamlining CRISPR workflows in ways we never thought possible. Imagine AI-powered robots handling the entire process, designing guide RNAs, running experiments, and analyzing the results. That's right. AI and robotics are making high-throughput CRISPR screens not only faster but more accurate. This means larger-scale studies can now be conducted with fewer errors and in a fraction of the time, pushing the boundaries of genetic research and helping us reach new breakthroughs faster than ever before. It's clear that AI and CRISPR are a match made in scientific heaven. By enhancing the precision and efficiency of genome editing, AI is helping ensure that we hit the right targets and avoid potential off-target effects. It's also playing a key role in understanding how patients might respond to these genetic edits, making treatment safer and more effective. Let's broaden the discussion. I invite Ryan to educate us on the different applications of CRISPR, De-Extinction, and social justice.
First off, I'll be talking about how CRISPR has the potential to recover lost biodiversity through bringing back extinct species. This idea was actually discovered before CRISPR technology in the 1900s through a variant of selective breeding, but it has the potential to be done much more accurately and efficiently with gene editing techniques. This seems to be more promising than other methods such as cloning and selective breeding. Giving a quote from Professor Tom at the University of Copenhagen, he said, if you know the sequence of the genome of your extinct animal and you have the genome of its relative, you can look for the differences and just use CRISPR to edit the differences. That's pretty much the basic idea of this. This is shown to be active through secure investors as well. A company named Colossal Biosciences, launched into 2021, explores this idea and has received $150 billion in investments as they try to revive species such as the woolly mammoth. There are also some concerns with this, such as there no longer being suitable habitats for some of these extinct species, and the concerns that we may disrupt existing ones by putting them back into some ecosystems. Another issue with this idea of CRISPR is biosecurity. So what are the risks associated with CRISPR technology through means like bioterrorism and unintended consequences of gene editing? And also, how can we mitigate these risks while promoting scientific progress? Alan Schaffer, the Deputy Undersecretary of Defense for Acquisition and the Sustainment, explained in his recent testimony to Congress that scientific advances are rapidly outpacing the ability to manage their consequences. Ease of access, low cost, and rapid developments to CRISPR technologies make this potentially very harmful, as essentially anyone can get their hands on this. Hands on information and the materials for relatively cheap. And converting these bioweapons can cause massive destruction over a large area for a relatively cheap price. The areas of highest concern regarding the development of bioweapons are actually the simplest applications, since they have been done before successfully. They're also the most dangerous. And some of these include recreating viruses, making biochemicals, and making existing bacteria more dangerous. And this has been proven in a Stony Brook lab study where they recreated polio using information online and ready-to-purchase materials. Another example is the mousepox IL-4 experiments, as they illustrate conceptually how pathogenicity or other characteristics of a microorganism could be changed in a harmful manner. In addition to the increased lethality from these gene additions, these experiments are concerning for potential implications to create a strain of the human variant of smallpox that could be resistant to the existing vaccine. Vaccines are part of deterrence by denial, since they make an attack with an agent unsuccessful, and thereby reducing incentive for an attacker. The concern is that advanced gene editing techniques can enable easier and more widespread manipulation of microbiologicals for use as biological weapons or bioterrorist agents. As time goes on and gene editing advances, we need to figure out how to be able to use this technology for good while preventing those with malicious intent from being able to do bad. What are some issues of equity and access to CRISPR-based treatments? How do we ensure that marginalized communities have access to the benefits of CRISPR technology and the potential consequences of exacerbating existing health disparities? This can include the grave underrepresentation of minorities in genomics research, which may lead to less effective and accepted CRISPR tools and therapies, and their anticipated unequal access to these tools and therapies in healthcare is widespread. Thus, although CRISPR's emergence has generated considerable excitement in the health sciences community, it also raises serious health equity concerns because historically, minority patients and populations have been persistently excluded from clinical research, innovations, and care, contributing to the current health equity gap by suppressing health benefits for groups experiencing the poorest health outcomes. Similarly, minority participants are minimized in genomics research, as most participants in genomic studies are of European ancestry, suggesting a future patterning in which minority populations will fail to benefit equally from CRISPR advances for improving health. Now back to you, Shahzad. Please inform our audience about the potential dangers and ethical concerns of CRISPR.
In this segment, we are going to explore the potential risks associated with the groundbreaking CRISPR technology. One of the biggest risks we face with CRISPR is something called off-target effects. Essentially, while CRISPR-Cas9 is designed to cut out or edit a specific part of the genome, it doesn't always hit the bullseye. Sometimes it makes cuts in places it shouldn't, leading to unintended changes in the genome. Why is this a problem? These off-target effects can cause mutations in genes that weren't supposed to be altered. Think about it. If we're editing genes in human cells, these unintended mutations could increase the risks of diseases like cancer or cause other unpredictable health issues. But that's not the only concern. Even if CRISPR hits the right target, there could still be unintended consequences in gene function. Genes don't work in isolation. They're part of a complex network that regulates our biology. So changing one gene can sometimes have a ripple effect impacting other genes or disrupting important pathways. This is especially risky when we're editing genes involved in critical biological processes. A small change might seem harmless at first, but it could disrupt normal cellular functions or trigger unforeseen physiological changes. It's like pulling a thread from a sweater. You don't always know how much it will unravel. Then there's the uncertainty around the long-term effects of CRISPR. While we know a lot about how it works in the short term, the reality is that CRISPR is still a relatively new technology. We simply haven't had enough time to study the potential long-term consequences of making genetic edits. This raises some important questions. Could these genetic changes lead to health issues that only show up later in life, or perhaps even across generations? The truth is, we don't fully know yet. This unpredictability makes it essential to approach the use of CRISPR in humans with caution and ensure we have strict regulations in place before diving into widespread application. While CRISPR is opening up exciting new doors in the field of genetic research, it's important to recognize there are still risks and uncertainties. From off-target effects to long-term unknowns, we need to proceed carefully as we continue to explore this powerful tool. Now, I'm going to talk about the highly complex and the most controversial topic, the ethical concerns surrounding the CRISPR technology. What exactly are the risks, and why are so many people concerned? One of the most profound ethical dilemmas arises when we talk about the germline editing. Germline editing means making genetic changes to eggs, sperms, or embryos, changes that can be passed down to future generations. Now think about the implications of that. We're talking about potentially altering the human gene pool permanently, and the ethical consequences are enormous. A big concern here is consent. Future generations who inherit these genetic modifications never had a say in those changes. This raises a question. Do we have the right to make such fundamental decisions for those who come after us? Another issue is inequality and access. Imagine a world where only the wealthy have access to gene editing technologies. This could lead to what some are calling genetic elitism, where the rich have the opportunity to enter their genes, while those without the same resources are left behind. The potential for genetic enhancements to widen social inequalities is a major ethical worry. Speaking of genetic enhancements, what about the fear of designer babies? Many are concerned that CRISPR could be used for non-medical modifications, such as selecting traits like intelligence or physical appearance or athletic ability. This opens up a whole new ethical question about what we as a society deem desirable, and whether that might lead to a loss of diversity or even the stigmatization of individuals who don't meet these newly created genetic standards. There is also the challenge of access and affordability. Another major ethical concern is the possibility of unintended consequences when we start releasing genetically modified organisms into the environment. CRISPR has the potential to alter the genes of wild species, and while that might seem like a solution to certain problems, it could lead to ecological disasters. Modified organisms might disrupt ecosystems, drive out native species, or introduce new forms of suffering into the environment. The potential for irreversible environmental changes is a significant ethical dilemma we need to carefully consider. Finally, there's a bright spot on the horizon with the epigenome editing. Unlike traditional CRISPR gene editing, which involves cutting the DNA, epigenome editing is reversible and cost-effective. Instead of altering the gene itself, it restores the gene's proper function, making it a safer and potentially more ethical approach to treatment. Researchers at Stanford are working on clinical trials in this particular area, which could provide less risky alternative to traditional gene editing. While CRISPR technology holds incredible potential, it also raises a host of ethical concerns that we cannot ignore. From consent and inequality to genetic privacy and environmental risk, there's so much to consider as we move forward with this groundbreaking technology. Now, I would like Josh to talk about the 2018 controversy.
In November 2018, Dr. Hai Jiankui made a stunning and controversial announcement: the birth of the world's first genetically modified twin girls, Lulu and Nana. He used CRISPR technology to edit their genes, specifically targeting the CCR5 gene, with the intention of making them resistant to HIV. At first glance, this seemed like a revolutionary achievement. However, the reality of this experiment turned out to be far more complex and deeply unsettling. The CCR5 gene is crucial in allowing HIV to infect human cells. Disabling it may appear to be a plausible solution. However, this experiment quickly became the center of one of the most significant ethical scandals in modern scientific history. One of the most serious concerns surrounding this experiment was the issue of informed consent. It had been reported that the parents of the twin girls were not fully informed about the risk associated with the procedure. Consider this: the genetic makeup of their unborn children was being permanently altered, yet they lacked a complete understanding of the potential consequences. In scientific research involving humans, informed consent is a fundamental requirement. Participants must be fully briefed on both of the risk and potential outcomes of the research. In this case, the parents were not provided with the necessary information. This represents a major ethical violation. Further complicating the matter was his blatant disregard for Chinese regulations. China has clear guidelines for research involving human embryos, including the 2003 Ethical Guiding Principles. They explicitly prohibit research on embryos beyond 14 days of development. Implanting these modified embryos into human uterus is strictly forbidden. Yet, this is precisely what he did. To make matters worse, the entire experiment was conducted in secrecy. Standard ethical review boards were bypassed, and regulatory oversight was completely ignored. It was a covert operation, revealed to the world only after the procedure had already been carried out. The global scientific community reacted with an alarm. CRISPR technology, while promising, is still in its early stages, and there is insufficient evidence to ensure its safety to use in humans, particularly in embryos destined to develop into full-grown individuals. Premature application of this technology can lead to unintended and potentially harmful consequences. For instance, one of the twins did not even receive the intended genetic modification completely, raising concerns about potential off-target effects, where CRISPR causes unintended genetic changes. Scientists also worried about the long-term health implication of the twins. Did this experiment expose them to unforeseen health risks by making changes we don't fully understand? This case sparked a broader debate about the line between therapeutic gene editing and human enhancement. Editing the CCR5 gene to prevent HIV might be considered therapeutic, but what if these changes made the twins more vulnerable to other diseases, such as West Nile virus? This incident had far-reaching consequences. Scientists around the world recognized the urgent need for clear guidelines on gene editing, especially in experiments that involve human subjects. It also highlighted the necessity for an international framework to ensure ethical use of these powerful technologies. And now we have Ryan with CRISPR's patent battle and physiological and social impacts.
Now I'll be talking about the ongoing heated dispute between UC Berkeley and the Broad Institute over the rights to CRISPR technology. It is a high-stakes legal dispute over the rights to CRISPR gene editing technology. UC Berkeley, led by Jennifer Doudna, and the Broad Institute, led by Feng Zhang, are the key players to this legal struggle. UC Berkeley was the first to file a patent application, but the Broad Institute was awarded key patents due to filing with a faster process. The legal fight revolves around who has the rights to the technology for different applications, such as in eukaryotic cells of plants and animals. So here are the key implications. The Patent Trial and Appeal Board, or PTAB, initially ruled in the favor of the Broad Institute, leaving UC Berkeley to appeal. The dispute has broad implications for research, commercialization, and future innovations in gene editing. This battle highlights the challenges of intellectual property in the rapidly evolving field of biotechnology. The ongoing appeals and future developments include multiple rulings and appeals throughout the past of the trial, with many more to come, and each side has claimed victories in different aspects. The final outcome could shape how CRISPR technology is accessed and utilized worldwide, affecting both academic and commercial entities. CRISPR technology, particularly when applied to human gene editing, raises questions about personal identity and psychological well-being. Changes to the genetic makeup of individuals, especially when made at the embryonic stage, could impact how people view themselves and others. Ethical concerns also come into play, as altering the mind-body connection could lead to unforeseen psychological consequences. Human enhancement and societal expectations. The potential for gene editing to enhance human traits such as intelligence, physical abilities, or appearance could alter societal expectations. There is a concern that such enhancements might lead to increased pressure on individuals to conform to certain ideals, potentially impacting the ideas of self-worth and mental health. The psychological effects of living in a society where genetic enhancement is possible could be profound, leading to new forms of inequality and social dynamics. Procreative beneficence and its implications. The concept of procreative beneficence suggests that parents may feel a moral obligation to enhance their children through gene editing. This could lead to a shift in parental expectations and affect the psychological development of genetically edited children. The societal pressure to create the best children could have far-reaching effects on mental health, identity, and family dynamics. The last topic I will be discussing: common myths associated with CRISPR technology. CRISPR can be used to clone humans. That is not true. CRISPR is a gene editing tool and not for cloning organisms as of now. Personalized vaccines using CRISPR. CRISPR technology has allowed us to make personalized vaccines to an individual unique genetic profile. This method could enhance vaccine effectiveness and reduce side effects by targeting specific antigens or mutations relative to each person. While still in its early stages, research into CRISPR-based personalized vaccines show promise, particularly in making more effective cancer vaccines and improving responses to infectious diseases. This will potentially revolutionize how vaccines are designed and administered to people. Myth 1. CRISPR can instantly cure all genetic diseases. In reality, CRISPR is not a magic bullet to diseases. While it holds promise, many genetic diseases are complex and involve multiple genes, making them challenging to treat with CRISPR at the current stage. Myth 2. CRISPR is 100% accurate. In reality, CRISPR is precise, but not perfect. Off-target effects are a real concern now, and scientists are still working on improving its accuracy. Myth 3. CRISPR is only for gene editing. In reality, CRISPR is a versatile tool that can be used for gene regulation, diagnostics, and even population control through gene drives. Myth 4. CRISPR is safe for use in humans today. In reality, CRISPR is still in the very early experimental stages for human applications. It is not yet fully safe or approved for widespread use, especially for germline editing. And lastly, myth 5. CRISPR is always permanent. In reality, not all CRISPR edits are permanent. Some can be designed to be temporary or reversible depending on the delivery method. Finally, back to Prisha to give us a sum up of what we've discussed today.
So that is all we've got for you guys today. Through this episode, I think we can confidently say that CRISPR technology can greatly benefit us in the sense that it allows us to edit and restructure the genes of the next generation. But this can only happen if we follow rigorous safety protocols, ethical guidelines, and maintain transparency with the public. Thank you so much for listening in today. Don't forget to hit the follow button and the little bell next to it. And if you want to further educate yourself on CRISPR technology or any of our previous episode topics, please feel free to check out the resources page. Also, if you have some time, please don't hesitate to give us some feedback on this episode. We will see you guys in the next episode.